water-related biodiversity monitoring methods with three of our speakers who are here. And one of the main topic of today will be by focusing on one prime feature, which is DNA. So now I'll leave the floor to our expert group leader, Sarah Iman, to showcase the expert group and the three speakers. Thank you.
Thank you very much. You can go to the next slide, please. I wish you a warm welcome again to our third webinar from the Water and Biodiversity Group. Shortly again, our goals to enhance the capacity of key actors in the water sector to actively combat ecosystem degradation and biodiversity decline. To that extent, we have sort of three main things we're working on: evaluation and identification of knowledge gaps, support mainstreaming of existing biodiversity and indicators and tools, and also looking at a framework for the development of these water-related biodiversity in different contexts. This webinar, as already Tiffany said, focuses on eDNA and how to use that effectively for monitoring water and biodiversity. It is therefore mainly on the second and third bullet. It is really about showing you new methods or extended use of existing methods, to support the use of these methods more commonly throughout Europe. To that extent, we have three speakers for you today. First, we have Professor Florian Leese, professor at the University of Duisburg-Essen. He is going to take us to the good parts of eDNA measurements, which have made so much more possible in monitoring biodiversity in water, but also is going to show us what the limitations are, how to deal with that, and the challenges that are currently occurring because of it is starting to be used much more and more, and that makes it we have to think about how do we want that to be used. So he's going to show us a way to much better and more efficient use, even beyond where we are now. Dr. Heike Zimmermann is going to focus on another part of eDNA measurement. We are going to look at the sediment eDNA. That allows us to look beyond the existing time series that we have now. So you have measurements for a short period, but you can look in the sediment and look way back. You can look at natural variations that have occurred in the past. You can look at the influence of climate. And in that sense, you can establish a much more robust baseline where you can go to and where you are coming from. So that's also a really improvement of the possible use of DNA. And then finally, Dr. Hetty Kleinjan from CBDO, she's going to show us a more newer method, I guess, looking at the genomic changes of periphyton within the eDNA measurements, which can really tell something about the response of biodiversity related to water quality changes. So that is another new method. And with that, I think I would like to leave the floor again for a few questions to Tiffany, because we really are interested in who you all are who are listening to us, and maybe get some extra information from your own experiences. And after that, we will go straight on with Florian. Thank you very much.
Thank you, Sarah. So here you have the Menti code and the QR code, just to, as Sarah said, get to know each other just a bit. So if you can, and I will also share the responses with the Mentimeter. Share.
People are signing up, so I guess we will get
Yeah.
the replies.
So I guess we can start with the first question. What type of function do you have and which type of organization do you represent? It's an open question, so we will just see all your responses. Engineer. Okay. Very diverse audience for now.
Oh, that's good.
Yeah.
At least I hope you will all connect to others again. A lot of research still.
Yes.
Yeah.
Okay.
Thank you so much.
Yes. We can go to the next question now.
Mm-hmm.
What is your relation to today's topic? It's one word. It will be a cloud one. Ah, yeah.
Yeah, so everybody is around monitoring water for purposes of framework directive or management, sampling, monitoring. It's around the thing, all around the same topics.
Yes.
Also more sediments people. I hope after this maybe there's more people going to be interested in the sediments as well.
Mm. Bioindicators. Citizen science is also important. Okay.
Oh, nice. Yeah, citizen science as well. Yeah, just go to the next one, I guess.
Yes. We have a last question before starting the presentation. Have you already been using DNA-based method to monitor water-related biodiversity? Mm-hmm. We have an expert in DNA, 2, 3. Let's- Some people are here to learn and would like to give it a try, so that's nice.
Yeah, it's diverse. It's from left to right.
Perfect.
Thank you.
Thank you all for responding to the Mentimeter. And now I can give the floor to Florian Less, who will start with his presentation.
Perfect. Can you confirm that you see the slide?
Yes.
Perfect. Just 1 second. I need to arrange the Zoom bar that it's not blocking my view. Okay, yeah. Thanks for having me here, and I'm really happy to start this interesting webinar session off with probably a bit more overview talk. And looking at your feedback from the Mentimeter, quite a few said, no, we haven't used DNA or eDNA-based methods, but we would try. We would like to try. And I hope this talk in particular is interesting to those of you that are planning to use DNA-based methods. First of all, why am I so much pushing forward to that. I really see the environmental issues we have, and I realize that seeing what happens on the planet is a challenge because, I mean, in particular, when it comes to biodiversity, right? We are looking at systems which span a huge number of, or a huge degree of order of magnitudes across size, right? From smallest viruses, bacteria, then to planktonic organisms, up to, in Germany at the moment, we have a whale that got stuck in the Baltic Sea. So then it really starts to become huge. So that means already with individual specimens, we have orders of magnitude which are huge. And then we move up to ecosystems, whole catchments. So then we really have to deal with thousands of kilometers sometimes, or of meters, thousands of meters we need to study. And the current system is not fit to do that because of this scaling issue. But we really need broadly applicable, comparable, and scalable approaches that are implementable not only in the rich global north, but ideally all around the globe to inform us about changes of biodiversity. So the vision that many people, including me, have is, why can't we have something like weather stations for biodiversity change? This is what we have in terms of the large weather stations, not the small ones. This is the large World Meteorological Organization weather stations. And these are important to help us forecasting how the weather changes and whether we are walking into catastrophes. So whether there needs to be a certain warning that some areas need to be evacuated because of a catastrophe coming. If we would ask ourselves how much of real-time data is currently generated for biodiversity monitoring, the answer will be quite sad, because not a lot. Our tools are not able to automatically and fast, ideally real-time, generate data. But we need that. Biodiversity information through space and time, comprehensive and reliable, and close to real-time, ideally. So the point is, this is one out of many tools. I don't want to convince you that only we should do DNA or eDNA-based methods. I think we have a great set of tools already at hand, but I think we need to complement it with DNA-based methods in order to address the scaling issue and to become quite efficient and effective from micro to microbial organisms. I often say DNA and not eDNA approaches because we also deal with analyzing specimens that were collected, for example, in a trap, which by definition would not be eDNA. What I would like to stress, in a way, it's all semantics, right? When does environmental DNA start and what is a bulk sample? What we are interested is using DNA methods to assess what lives in a certain environment, whether the specimens or just an environmental sample with the specimens and traces within. I will mostly focus in my today's talk about a method that's called metabarcoding. So either you collect your organisms in traps or you collect water or sediment from a stream or from the marine realm. Then basically you need to extract the DNA from this sample and then you amplify the DNA that's contained within that sample using a polymerase chain reaction in order to measure which species are in there through a certain well-defined standardized genomic region, which is called the DNA barcode. This can then be read out on a sequencer, and we have a fantastic array of sequencing machines that do the job. And then there's some magic that happens with bioinformatic analyses, but also there we have good solutions. And then we end up with an overview list of sequences that can be assigned to species, and we see what lives. So it's basically occurrence data in space and time. And the method is not new. In fact, microbiologists are using this for over 4 decades. And we, as the microbial world people, we started to identify that only about 22 years ago when the concept of DNA barcoding emerges. But it's not really new. It's innovative, but not anymore new. already back in 2019, the Joint Danube Survey, for example, used DNA and eDNA-based methods, actually look at those chapter numbers here, to study biological quality elements across the Danube. And I would like to highlight this very, very nice overview graph, which shows from the source of the Danube to the mouth, the presence of various fish species, which are on the y-axis. So just by filtering water, the experts could assess which species were there, and the diameter gives the relative reed abundance. Quick side note, you're not only stuck with water. We are doing the National Monitoring of Insect Biodiversity as part of the Long-Term Ecosystem Research Germany program. And we have 75 traps all across the country. And just within 2 years sampling, we were able to track 31,000 insect species. Just as a side note, for Germany, only 33,000 insects are described. So with that fantastic sample collection and DNA-based assessment tool, we can assess a broad, almost the majority of the insects occurring in Germany. And the costs, just the laboratory costs, are quite low, 10 euros. Then, of course, you need human labor cost, and then easily it can be 100, 200, 500 euros. Still, it's affordable, at least compared to traditional assessment methods. So big advantages, why I think the strength is so unique of DNA, and here now, especially the eDNA methods, where you just collect a water sample. You can, from this one water sample, that's 1 liter of water we collected from a river nearby for, in the context of 1 year. Every 2 weeks, we collected a bottle of water, and basically we could assess from the microbes to the macrobes, even to the terrestrial mammal species, what occurs where and how does diversity change. For example, the fish diversity through time, bird, mammal, freshwater invertebrates, terrestrial invertebrates. There you see that you have quite seasonal trends in there, or the diatoms. And that is really fast and affordable. It doesn't cost a lot. And from one sample, you have across the tree of life data. It's, however, not the holy grail. You definitely have lots of false negatives and sometimes a few false positives in there. All methods have that. So, and you can even go back to the future. In Germany, for example, we have this fantastic environmental specimen bank. Germany was extremely clever with respect to this one. Back in the 70s, they bought lots of liquid nitrogen tanks, and they store various environmental matrices in there, amongst them sediment from rivers every year. And from that, we can now extract eDNA and see how, for example, fish diversity across the major river basins changed. Or what are the winner and the loser species? For those of you that know fish quite well, of course, the gobies are here on the top, many of which come from the Black Sea region. And then we have a few ones which are really the losers at the moment. And the data is very compatible and consistent with the traditional red list monitoring. So this led us to propose it is a method that is scalable. Especially eDNA from filtered water is a method that, due to the simplicity, can be scaled up globally. Citizens can apply the method because it's so simple, accepting that it's not a 100% holy grail method, but a very strong one. So if you're interested, Florian Altermatt and a couple of colleagues, we wrote this paper how to scale that up globally. And it's really not, I would say, an idealistic view. Many pioneering projects from all around the globe showed it's possible. I just would like to mention, for those of you that have not worked with eDNA, we should get rid of the thought you measure something with DNA and you measure something with traditional methods, that both things should match perfectly. So this is what often people think. If you have a box here and you identify it once morphologically, then you do it with DNA-based methods, it should 100% overlap. No, it won't. Those samples have parasites, they have gut contents in there. And also here misidentifications can occur. With DNA methods, you may have incomplete reference databases. But even in a perfect world, you would not find the parasites or the gut content through a traditional method. That's not a bug. I would say it's a feature, and we should not try to make this 100% comparison. Especially also with eDNA, the method cannot detect some of the organisms that do not shed DNA. So we should just accept that there's a certain overlap, but that's actually quite comprehensive. And then, and this is also nothing new, the people in the hospitals do it all the time when they buy a new X-ray device, then you have to calibrate the new and the old machine. We can do the very same in biomonitoring and assessment by taking a new assessment method or an adjusted assessment method, use the European Commission's intercalibration guide developed by the working group EcoStat under the Water Framework Directive. And we did this for benthic invertebrates. And you basically see before that you had a slightly upwards biased assessment with DNA tools. If you use this, if you apply the proposed method afterwards, you have an intercalibrated, I mean, we are not allowed to intercalibrate, but we can show that it technically works. The official procedure needs to be done by the intercalibration group of the Commission. So this is why so many people, if we ask them, especially that's now the national experts on the Water Framework Directive under EcoStat, do you think DNA-based approaches can facilitate ecological status assessment in the context of Water Framework Directive? 22 responded yes. 1 no, 3 don't know. And that's the official national representatives. Are your water authorities willing to include DNA-based methods? 11 yes, 2 no, 13 don't know. Do cost issues play a role in decision making? Yes, they do. Just from Germany, I just received here our water economy newsletter, and we read here, we lack experts for traditional monitoring. So that's a big issue, and a cost issue also occurs. So that's some of the motivations here. So to end now with my talk, what are the key challenges? Key challenges exist, and we should very openly communicate them. One is barcode reference libraries. They are not complete, especially if we go to countries or in continents like Africa, global south, extremely species rich, but barcode coverage, even for fish species, underrepresented. However, this was just published a couple of hours or 1 or 2 days ago by a team around Florian Altermatt. We see that despite the lack of barcode coverage, the richness estimates actually are quite consistent, even between the quite old estimates that were done sometimes back in 2014 and reassessed, reanalyzed richness estimates by Florian Altermatt and his team, the lead authors Yan Zhang. So we also see that various different bioinformatic tools were used. The results are quite consistent. This means there's a lot of hope despite the need to further improve reference databases. Next point is the quantitative data are weak with DNA and eDNA metabarcoding data. We should not try to force it to a perfect quantitative method. It will not work. But we can use image-based methods and other approaches and complement these. This is my take-home message. Don't try to make DNA and eDNA too quantitative. It won't work. Use complementary methods. And the last point is we need now, and it's really time to do that now, write standards and stick to those standards. We are living in a wild west, increasingly wild west of methods that are being used. Great for research, very challenging to compare the assessment results. Fortunately, we now have at ISO level and CEN level a standardization working group, and it would be great if experts of you could join that group, because the challenge, just to show it here, join this expert group if you are interested. It's under Technical Committee 147. So to sum up, the future is now. The powers really methods are simple, scalable, cost-effective, and you get from one sample of water from bacteria or from viruses to vertebrates. But we have to now work on standards, quality assurance, work on completing the reference databases, and try to use different stuff for the quantitative aspects. Yes, with that, I just would like to mention the fair data aspects are being covered by various projects. Great progress made. If you're interested, these are projects dealing with that. And we look forward to soon having a European eDNA society that really will try to embrace all the experts working in the field to make sure that the Wild West will get tamed a little. Thank you very much.
Thank you very much, Florian, for your interesting talk. I think it was very good to follow for all different types of people. And I really agree with this complementary method. So I am happy that you emphasize this as well. So let's try to make it better at what it actually is good at, and do not try to use it for things it's not so good at, because we have other ways to do that. Then if there's already questions, you can put them in the chat. Otherwise, you can leave them for after the three talks. And I would love to give the floor to Heike now.
Hi, everybody. I'm Heike from the Geological Survey of Denmark and Greenland, and I want to thank you for inviting me to speak today about sedimentary ancient DNA, which is essentially environmental DNA from the past. And we can use this tool to define biodiversity baselines, or in other words, a reference stage or reference condition. So the question is always, what are good reference conditions? And no, my slide won't shift. Okay. Monitoring programs are very important in generating those reference conditions. And one of the oldest program was from the Marine Biological Association that was established in 1931. And for the Arctic specifically, we have, for example, the Greenland Ecosystem Monitoring, established in 1995, or the Circumpolar Biodiversity Monitoring Program from the Arctic Council, established in 2002. But if we look at observations of the Arctic average monthly Arctic sea ice extent, we see that especially in the timeframes covered or started by the latter programs, the Arctic is already affected by climate change. So we need to go a little bit further back in time. But it is not just enough to go to a stage pre-pollution or pre-industrial revolution. It is also important to be aware of climate anomalies. So, for example, the North Atlantic multi-decadal oscillation can have very strong impacts on the species diversity in aquatic ecosystems. So it depends really on the phases we're looking at, because within just a short amount of time, like decades, there can be huge regime shifts that affect the food web from protists to marine mammals. We know that there are very high uncertainties when we want to project future ecosystem conditions, but if we include paleo data, or for example, traditional or indigenous knowledge, we can actually strongly reduce that uncertainty. And therefore, paleoecology or paleo data is increasingly used in restoration ecology. And a big important aspect is that paleo data can help us identify ecosystems that are looking vastly different from today. And especially in Europe, where we have successional stages of cultural transformations, it is important to know or to avoid any misconceptions about what we believe is a natural ecosystem. So in my opinion, ancient DNA could be an important tool in assessing biodiversity. And for example, if you have an aquatic environment, you have different organisms across the food web, and they can shed feces, skin, or they, like the dead cells, can just settle down to the seafloor, and there they can get incorporated into sediments. So if we take a sediment core, we can look layer by layer how biodiversity changes over time. So the further down you go with the sediment core, usually also the further back in time you go. So here we see some strengths of the DNA method. So we can use this up to geological timescales. We cover all domains of life. We can use this method in high throughput, and we can actually link the sequencing data not just to taxa, but also to traits. And sometimes it can be used potentially quantitative, but it really depends on the method you're using and also the marker you're using. But of course, it has also some weaknesses. So for example, we cannot get direct phenotype information. We have gaps and errors in reference databases. We also have prokaryotic and fungal signals of species that could be alive in the sediment, so they can overprint actually ancient eukaryote signals. And then, of course, different labs can use different extraction methods, they use different markers. So there's really also a bias in, like if you want to compare data sets that were generated in different groups. And finally, we have a big risk of contamination, and this contamination risk accompanies you through every step of the lab. For field work, we can go, for example, and take lake sediment cores, and this is actually quite easy, especially in the Arctic, where you can use, for example, the lake ice in summer, in winter, and use this as a platform for coring. And in summer, you can just go out on a rubber boat, but for marine sediments, it's a bit more tricky. You have to go out on a ship, and then you get gravity cores that have to go through sometimes a kilometer or more through water column. So once we get these sediment cores up, we chop them in 1 meter long sections and take them back home, where we can open them under controlled conditions to avoid contamination. And you see that here I'm really covered up in a suit, and we even wear 2 layers of gloves to really make sure that we do not contaminate the ancient signals with our own DNA. And we bleach everything and ethanol everything and use sterile equipment to take samples from the inside of the core. Then when we have the samples, we bring them into our dedicated ancient DNA lab. And here you can see our brand new labs at GEOS, where we can do DNA extractions and, for example, PCR setups, or also library preparations for shotgun sequencing. But in this talk, I want to focus on DNA metabarcoding, and I'm glad that Florian has already introduced that, because it is a really simple method that is relatively cheap, and also because the reference databases are much more comprehensive than any other reference database that we have. And we can use PCR markers that are targeting broadly eukaryotes, but we can also target markers that are very specific for a certain taxonomic group. And here we used a diatom-specific RBCL marker. And for the difference for working with sedimentary ancient DNA is that we have to focus on very, very short markers, because the DNA is ancient and degraded and highly fragmented. So the shorter the markers, the better. In the next slides, I want to focus on 2 case studies where we looked at a sediment core from Northwest Greenland that is taken close to Upernavik. And in the first case study, I was comparing diatom sedimentary ancient DNA with the microfossil record. And as Florian already mentioned, here we see that it's really complementary. We found that almost half of the diatom species that we detected in the microfossil record are not even in GeneBank. But we also saw that the DNA is better at resolving, for example, pennant diatoms. Also, we see that RBCL has a higher overlap with the microfossil record than the V7 region of the 18S. So we really have to be careful how we interpret data and reconstruct our environments based on that. For dinoflagellates, we did the same, but here it is a little bit more challenging because of the dual nomenclature. So in these two pictures, you see exactly the same species, but we have the motile stage in the water column, and in the sediments, we find the resting stage or the cysts. And many of these cyst-teca relationships are today still unknown. Once we compared the DNA with the microfossil record, so on the left side, you can see the cysts, and in the middle, the motile stage that are affiliated with the cysts, and on the right side, you see the DNA. And we see that there's quite a diversity of cysts, but there are very few linkages to the DNA. So the cyst producers are really underrepresented in the cetaDNA record. But for example, Gymnodinialis were more diverse in the DNA record, and also we found several orders that do not even preserve as cysts, and those contain a lot of parasites or endosymbionts. So we have to be really aware of the biases of the different records, and do not just use sedimentary ancient DNA as the sole proxy for reconstructions. In our second case study, I wanted to show that we can link also sedimentary ancient DNA with trait data. So here you can see an overview of the functional groups that we could cluster, and this overview really shows the variety of the diversity responses that we could find. And here I want to focus on parasites and saprotrophs, which they are not found in the microfossil record, but they have the highest diversity. And we also found a high diversity among phototrophic floaters, which here are only the diatoms. For those, we actually found a strong negative correlation with our temperature proxy. So that means that diatom diversity is actually higher under colder conditions. And we found the same for pelagic fauna, but we see a different response for benthic in fauna, where, for example, we see that there's a negative correlation with our sea ice proxy. However, here we see a peak that falls into the same time frame where we see also a peak in primary productivity that has been exported to the seafloor. So we assume that the exported productivity has fueled benthic in fauna diversity. And also we see for benthic epifauna that there's like a pulse-like diversity pattern over time. And with spectral analysis, we found that there's actually a 1,500-year cycle that fits with dominant swings in the Arctic Oscillation. So the key messages are really to be aware of the biases and that different processes can drive the diversity of functional groups. And here we see that it was related to temperature, sea ice, primary productivity, and even the Arctic Oscillation. So all these processes mean that we really have to look at long-term paleo records for establishing baselines, and these should really include also the whole range of the variability. I was asked the question if sedimentary ancient DNA metabarcoding DNA can be also seen in applications for monitoring aquatic biodiversity. And I think that would be brilliant because we need to learn from the past. And this can help us answer questions like how to define the baselines for biomonitoring, restoration, or also preservation. Then what ecosystem state do we want to preserve or restore? It can help us find out, should we focus on historical, cultural, or rather natural ecosystems? But a big question, I think, remains to find out if ecosystems will respond the same way in the future as they did in the past. So for example, can we actually use phases like the Holocene thermal maximum as an analog? And for marine ecology, I think the biggest challenge is access, because ship time is very expensive, and there has been a lot of coring activity going on across the whole world over decades. So there are a lot of legacy cores, and I think here this requires only collaboration and the repurposing of archive materials. So this is really an opportunity. For lakes, there's of course easy access, and then I see some further challenges like the processing of samples. So if there's really low biomass, which is happening most of the time, unfortunately, then a SEDA DNA lab is required. So this could be a barrier, I think. Then also the natural state. So sediment records, especially lake sediments in Europe, might not go as far back in time as needed to find out what the real natural state is. And of course, as Florian already mentioned, we have this huge database gap, and also we have gaps in our understanding of the physiology, ecology, and the adaptive potential of species. So I really want to highlight that omics stands on the shoulders of taxonomists, and that we have to come out of the respective silos and really tackle this in a community effort. And with this, I want to thank you and my colleagues and the foundations. Thank you.
Thank you very much, Heike. I think you quite clearly showed also the difficulties, but definitely also the questions that we can answer using this method. Yeah, I wonder at what time scale this will be done, like in urban environments and things like that. But on the other hand, it will really help to answer useful questions like where should we try to get at all? What is feasible? What is useful? So no, I'm curious to follow this in the future. Thank you very much, and I would like to go to the last speaker of today, which is Hetty. She's sharing her screen already, so.
Do you see my screen in presentation mode?
Yeah.
Okay. Okay, well, thank you everyone for joining this webinar. My name is Hetty, and I will today present a project that I've been working on since the last couple of years. And in this project, well, this project is called Aqua Biosense Project, and in this project I study the community composition of aquatic biofilms, also called periphyton. And to do that, I use DNA-based approaches. And yeah, the concept of environmental DNA was already nicely introduced by Florian Leese. So yeah, environmental DNA, yeah, it's a matter of definition, but is DNA isolated from environmental samples as exposed to maybe DNA directly from individuals or individual organisms. It has several features that makes it an excellent technology to assess biodiversity, mostly related to the rapidity of the analysis and the fact that it's less intrusive. And there are two levels at which you can approach this eDNA concept. So you can either approach it from the type of matrix that you study, soil, sediments, biofilms, or water, or you can approach it from the taxonomic side and focus on the identification of different species from the different clades of the tree of life. In the Aqua Biosense project, as I said, we focus on biofilms and periphyton biofilms that are complex communities comprised of different organisms, bacteria, microalgae, but also microscopic animals. So periphyton, as I already said, it's another word for aquatic biofilms. There are biofilms attached to substrates immersed in water, either artificial or natural substrates. They are complex communities that cover different trophic levels, organisms coming from different trophic levels. So it includes primary producers, fungi, but also heterotrophic bacteria and some microscopic animals that use the biofilm as a food source. Because the biofilms are constantly exposed to the water column, and also because of their intrinsic characteristics and their matrix components, they behave kind of a sponge, let's say, and they can accumulate and absorb components that are present in the water, including pollutants, micropollutants, but also other microparticles that are in the water column present. And because this periphyton is continuously exposed to the water, its community composition is also constantly evolving in relation to the change in the water composition. And we use this characteristic to study the microbial biodiversity in relation to the contaminants that are present in the water. And the project aim is to use the changes in the biodiversity that we find in the periphyton as a global indicator of water quality and ecological health, and use this as a bioindicator to complement existing or more traditional methods that are based on counting the individuals manually. Yes, like I said, so what we aim to do is to move from more conventional methods based on assessment on physical characteristics to identification of biodiversity based on DNA fingerprints. And the DNA fingerprint allows us to have a digital signal and creates a way to make the biodiversity assessment automatable, and also in a sense it helps to increase the frequencies of analysis that you can carry out, and thus creates a higher resolution of data over time. So our methodology, it covers basically the whole chain from sampling to periphyton collection, extracting the DNA, the sequencing itself, and then also the bioinformatics related to the identification of the taxa that are present in the samples. So we have a field campaign, and we use this data to calculate a DNA-based bioindicator of water quality. And finally, we hope to develop a methodology that is applicable in the field. Shortly on the sequencing technology that we chose to use for this project, it's nanopore sequencing. It's third generation sequencing technology, and its main advantage is that it's a portable device and it's rather cheap, but it has the difference with, for example, Illumina sequencing, that it is based on long reads and not on short reads. So there's a difference in methodology there. In the project, we use the 16S and the 18S barcodes, and to assess prokaryotes and eukaryotic organisms present in the periphyton. So the field campaign, it existed of the installation of passive samplers in and different rivers with different water qualities. The rivers, they were exposed to different types of pollution, so they have a pollution gradient from up to downstream. We also included some smaller streams for which we know that the water quality is better. And we also included one pond as a stagnant water mass. There was a monthly sampling campaign where we manually scraped off the periphyton from the passive samplers that we installed. In parallel, we characterized the water for the physical-chemical characteristics, and we calculated water quality indices. We did this for 1 year to also capture seasonal effects. And among the sites, there were also several sites that are officially with official locations for the Water Framework Directive monitoring plan. And this is how it looked like. So some photos to give an impression of how we installed these passive samplers and how the biofilm was attaching to those samplers. And then the next step in our projects was to calculate the bioindicator and to couple pressure and pollution pressure with impacts on community level. So for that, we used the physical-chemical data from the water quality, and we coupled that to the biodiversity data. So we calculated what different water quality indices. There are really a lot of them. So for now, we have chosen only those few, but there are more. And we coupled the water quality indices and the classes with the presence of specific species in the periphyton community. And these species we call indicator species. So it's kind of looks like this. So on this table, you see the location. Each location is classified according to the water quality that it is, that we measured. And then for each site, we also have a community profile with bacteria and microeukaryotes that were identified in the periphyton. And then we coupled this information together, and we identified organisms that were either unique to the water quality class and also organisms that were unique to the water quality class, but perhaps not present in each case. And from that, so this is how the water quality indices look like for all the sites that we monitored. So what you can see here is that dependent on the quality index that you choose, the water does appear to be very have different quality. So the two at the left, they are more their indices more calculated on physical-chemical parameters. And we do not see very many sites that are classified as very bad nor as excellent. But when you look at the trophic state index, for example, we have many more sites that are classified as hypertrophic or eutrophic. So this is already one first observation that we made. And then for each of these water quality classes, we identified bacteria and also eukaryotic species that correlated with these water quality classes. And when we look at those results, we can see clearly that some of the classes, even though, for example, there were many sites with moderate water quality, we didn't identify any indicator species. But While also, for example, another observation that we made was that some water quality indices do not give any indicator species at all. And yeah, so we can conclude that it really is indice dependent on the type of indicator that you identified and how many there are. And if I go into more detail now, for example, for the one quality indice, which is called the DCE, so Water Framework Directive SEC-O, which is the physicochemical quality of water. We see that there were, for the water quality classified as bad, we identified 2 bacteria with the most statistical significance, and also on the eukaryote part, we identified 14 species for good water quality class. And those for the good quality class, they were, yeah, maybe as expected, mostly diatoms, while in the bad quality water quality indice, we identified mostly some bacteria. When we look at the trophic state index for nitrogen, we see a whole different picture. So we have some species identified for oligotrophic environments and mesotrophic environments. They are mostly bacteria. However, we do not see any eukaryotic indicator species. But for hypertrophic environments, we do see both bacteria and also eukaryotes, including ciliates and green algae. And the same again, but for the trophic state index using phosphor, where we have many indicators, mostly indicators for eutrophy and hypertrophy. And we observed that, again, for eutrophy, we have bacteria and diatoms that are indicators of this water quality. And for hypertrophy, we again see the ciliates and the green algae popping up. So in summary, we can conclude that based on this first analysis that we did of the data that is available, we identified for now 52 organisms that correlate the most strongly with the different water quality classes. However, this data set is being analyzed as we speak, so it will be expanded and also be enriched in the next couple of months. But I think it demonstrates already now that the periphyton can, or the periphyton community can be a useful matrix to identify new species and also to study the impact of pollution in the water on the biodiversity present in this matrix. And yeah, as I said, this list of bioindicators is now available. And there are, of course, still tasks that are in progress, such as enriching the dataset with more sites. Also, we'd like to include macroinvertebrate data, and perhaps more importantly, to confirm the ecological context of the identified species using bibliography, but also existing databases that are used to calculate, for example, the diatom index or the global biodiversity index. Yeah, so we'd like to align also with the regulatory observations that are carried out in the Water Framework Directive, and automate this method eventually. So my take-home message would be that periphyton communities do show a clear shift in response to changes in water quality, and that they can indeed be used to monitor biodiversity and to identify bioindicator species. And the added value of this method for effective water management are, which is already said before, related to the fact that you can monitor multiple taxa from the different parts of the tree of life in one analysis. You can possibly do it in fields with the sequencing that we used, and there's minimal training required to perform this analysis. And altogether, it allows you to take action more quickly whenever a disturbance occurs. And I want to close with those 3 open questions that I'm sure that have popped up in your mind during this speech, and that can maybe help to feed the discussion. But the question would be whether this can be used as an early warning system. What about the baseline, and are those indicators ecosystem specific, or can we also transfer them to other cases? And with that, I end my presentation, and I thank you for your attention.
Thank you very much, Hetty, for your nice talk on a different way of using eDNA for indicating water quality. I think it's nice that we finished just in time to have some of the rest of the questions. Tiffany, do you have them ready?
Yes, I do. Alors.
So we go back to the Mentimeter because it's the only way that you can actually let you talk. If there's anything else that you want to say as participants, please leave your messages in the chat. We will get back to them. Yes. Either straight away or at a moment soon to come.
Perfect. So our first question, what water biodiversity application would you use DNA-based methods for?
What is the question that you want to answer mainly? Is it an open question, this one? Yeah, I guess so.
Yes.
So you will have to go back in your Mentimeter again. Sorry about that.
Before. Okay.
So biodiversity impact studies, that is to say the impact of things that happen on biodiversity, or the impact of measures that you take on biodiversity, or both? I always wonder whether it is in a positive sense to show effects, or if some disaster happened and you want to see what the consequences were. So that's an early warning system that requires a really quick analysis. If either Florian or Heike or Hetty has to say anything about that, please go ahead. You can react on this, speakers. Is it quick enough, the eDNA, to use as an early warning system yet?
Maybe on that. It often has been sold or is still being sold as very fast and very cost effective. And definitely it has the potential. However, the samples we get sometimes take up to 8 weeks until we give results out. So this would require that you really have the infrastructure in place. But yes, definitely it can work, but it requires also who will pay for the early warning system.
Yeah.
And also, it's much easier to monitor one pathogen as opposed to whole biodiversity.
Yeah, I understand that one. Yeah, so you can't actually do quick analysis, but then you have to know what you're looking for already.
So this being done in Germany, for example, with now the harmful algal blooms, we had in the river Odra between Germany and Poland, this big catastrophe. And now, for example, for this microalgae, it's Primnesium, there's an assay. So you could quantify also the abundance of this harmful algae individually. That's really a fast one. And there are techniques like LAMP or CRISPR-Cas-based methods, which can be very sensitive and fast.
Thank you. Anybody else has a reaction on everything that's here in the title? Sorry, my computer's going down.
I like this remark on the dashboards, but it was also already mentioned by Florian in the introduction that it would be like the ideal output in the end, to have a dashboard where we visualize the biodiversity states in semi-real time or real time.
And I would really love to also comment on this support to traditional methods. So I really think we have to make sure that while you accept that novel stuff comes, we really must be careful not to now kind of abandon the old stuff, right? There's so much good there. And I'm a bit afraid that we will move towards lots of generalized ecologists with some taxonomic knowledge, but we lose the experts. We really need to have the taxonomic experts. We don't need ecologists that can use AI identification methods or keys. We need taxonomic experts, and we have to make sure that we have a revolution by getting more of really the nerds, the experts that are experts for specialized groups. Everybody can identify birds, right? We don't need more there.
A bird from a fish, yeah, that would work, yeah. No, that's a very good one. In Dutch, we would say, don't throw away the kit with the bath water. Like, if something new comes, don't throw away the old things. We just keep them and improve it. Thank you for all these reactions. You're going to the next one? Yeah, you are.
So what would you need to start using DNA-based methods to monitor water biodiversity?
I think this is specifically a question to the people that say, I would like to try, but I didn't do it yet.
Maybe we change also some minds.
Sorry?
Maybe we also change some minds for the ones who weren't sure.
Yeah. More acceptance from regulatory bodies. So that's EU and national, I guess. Guidelines, well, they're being developed, Florian. That's what you just showed, basically. Can they be found already somewhere?
Yes, the ISO standards, they can be found, and guidelines exist, probably 100, but
yeah, that's the issue, which guideline to choose from, yeah.
Yeah.
And the ISO standards, they are in your presentation. Is there a link as well?
So I can share a link. It's ISO Technical Committee 147, Subcommittee 5, Working Group 13. And standards take time, 2-3 years, at least 2 to 3 years until they appear, because it's a consensus-based process. So the first is now almost done. 3-4 more are about to.
Oh, that sounds good. Yeah, if you can share a link, because I'm not able to remember this whole line you are doing very fluently. Heike, did you want to add something?
Yeah, actually, I want to talk about ISO standards or guidelines in terms of sedimentary ancient DNA, because here we look at sediments, and often, especially in marine sediments, the sediments are very differently. They have different mineral compositions. Sometimes there are huge amounts of inhibitors. So that's actually why we have so many different extraction methods, because sometimes one method works for a variety of sediments or layers even, and then for some sediments it just doesn't work at all, and then we have to shift, and so this creates this a little bit of a mess, I think, if you want to compare, for example, marine sediment cores from Greenland to what you find maybe in the tropics or in Antarctica, and then it gets a little bit more complicated, I think. But I think for maybe lake sediments, it could be maybe easier. I don't know. I have not that much experience with that.
No, I understand. Thank you. I see money a few times. Of course, we need money for everything. Reference database, standardized methods. It's a lot of things. And is money related to acceptance from regulatory bodies? Or is it not? Did anybody say something about that? Or chat if it's someone else?
I mean, it's really a key point we see with Water Framework Directive, that people say in Annex 5, Water Framework Directive says you have composition and abundance. DNA-based methods don't give you abundance. We are not allowed to use it. So this needs to be opened, as often it has been shown that even without abundances, sensu stricto, the methods really work smoothly. And Ecostat is working on it.
Yeah, I wanted to say, I know that Ecostat is working on this.
Yes.
But I wonder how quick it will come through. Let's see that.
If I may just say something, I think when you send message to the chat, it's only for host and panelists and not for the participants. That's something you can modify. You can put everyone so that
yeah, I tried, but I cannot do today. I could do that before.
I will take it back. This is the link of Florian.
Oh, yeah, I see it. Yeah.
And I think we should wrap up now. It's time for everybody.
Yes. I think there was just the last question. Yes. If we can get your contacts and information.
So please leave us an email if you want to receive anything or any other information you'd like to share. Or we are working on a white paper that also contains a lot of the information that you heard today. So if there's anything you want to add or you want to share, please let us know.
Yes.
And then I would, from my side, wish you thank you very much for your presence here, and thank the speakers very much for their interesting and new topics that they shared with this group. Is there anything you want to add, Tiffany, or any of the others?
I think the only thing I would add is to stay tuned for other webinars from our expert groups, and one will be held in May or June, would be the fourth and final one. Yes, from our Water and Biodiversity expert group.
Thank you so much, then.
Thank
you to all,
and
have a nice week.
Lunchtime.
Yes, thank you.
Bon
That's
appetit.
very interesting. Thank you so much.
Thank you.